Hi there. So today we're going to talk about the respiratory system, and this is a recording for Biology 233 during the summer of 2018 at PCC Southeast. So we ended last term partially by talking about the cardiovascular system and about blood, which is the main way that gases, both dissolved and carried by molecules, get moved around the body. So the primary goal of the respiratory system is to gather up gas molecules from the environment and dump gas molecules into the environment. So the gathering component means collecting molecular oxygen, so O2. And I want to remind you, because people still got this wrong on the last exam, and the practical O2, think back to 231, this is two oxygens covalently bound together. So that is one atom of oxygen, and here is another atom of oxygen. So we're going to keep circling back to this chemistry stuff. It's really important that you understand it, and I'm going to keep asking you about it until I know that you've got it. So molecular oxygen, that's what that means. So cells use this to oxidize food for fuel and make ATP. And one of the waste products is carbon dioxide, so a carbon bound to oxygen. The other waste product not mentioned here is water. And you'll learn all about that chemical reaction when we get to nutrition and metabolism. So the respiratory system is designed to exchange the molecules across a thin membrane, in the lung. And the cardiovascular system is designed to transport those gases, either carried around by a protein like hemoglobin or dissolved in plasma, both to and from the lungs and to the tissues. So failure of either system results from rapid cell death due to hypoxia. So hypoxia is low oxygen levels. And hypoxia in the tissues, can result from ischemia, which is not enough blood. So that would look like a clot traveling to the left anterior descending coronary artery and causing ischemia of the downstream myocardium in the ventricles. But it can also just be caused by obstructions to the airway or anything that impairs the ability of the blood to carry oxygen and drop it off where it's supposed to go. So a little bit later we'll talk about some ways that can happen. One of them is carbon monoxide can take the place of oxygen on your hemoglobin and make it really hard for you to exchange gases. So some general aspects of the respiratory system. You've got airflow that is designed to get air to your lungs. And within the lungs, you have bronchi. These are the largest tubes. Bronchioles, which are smaller. And finally, alveoli. These are dead-ended small sacs. And they are made of simple squamous epithelium. So we're going to keep bringing principles from 231 and 232 back into this stuff because, of course, all of it is ultimately related. And in order to show me mastery of these concepts, you need to show me how the concepts we're learning in this term relate to the concepts that you should already know from previous terms. So that's why there's that requirement that you have to pass one to take the next one. So within the respiratory system, there is the conducting division. And this is the area of the respiratory system that is designed only to conduct, meaning get air to the place where gas exchange occurs. So no gas exchange, meaning no collecting of oxygen or dropping off of carbon dioxide, is occurring within the conducting division. Now I'm going to change pen colors here for the respiratory division. So red is going to represent oxygen. So respiratory division, this is where gas exchange is occurring. Oopsie. There we go. So those are divisions of the respiratory system that are concerned with whether or not molecular gases are being exchanged. But you can also divide the respiratory system anatomically. So these two are dividing based on function, which I'm going to abbreviate as FN. And these two are dividing based on, structure. So the upper respiratory tract is what it sounds like. It's up. So these are organs in the head and neck, and the nose down through the larynx. So basically your face through your throat. Once you get to the end of your neck where it connects to your trunk, the lower respiratory tract is the organs of the thorax, including the trachea, through your lung tissue. And that includes bronchi, bronchioles, and alveoli. So we're going to start with describing the anatomy of the upper respiratory tract. And we're going to work our way down to the lungs. And then once we get to the lungs, we'll talk about the physiology of breathing. So the nose, you guys all have one. You know what that is. So it's the thing that looks like this. I don't know why that weird line showed up, but there it is. So there's a nose. So functions of the nose. Warm, clean, and humidify inhaled air. So how does it do this? Well, you have nose hairs and they trap particulate matter because they're covered in goop, which is mucus. So hairs and mucus are good for that. Of course, we learned in sensory stuff that they also detect odors. And it's a resonating chamber that amplifies the voice. So this is why when you have a cold or you hold your nose shut, your timbre of your voice changes. So that's right, important for communication. There are both bony and cartilaginous supports for the nose. So the superior half are the nasal bones medially and the maxillae laterally. So you might think of the nose as only being comprised of the nasal bones, but those are really just the bridge of your nose. On either side of those two bones, there are upward sloping areas of your maxillae, behind which are the maxillary sinuses, and that is contributing to your nose shape as well, particularly the bridge of your nose. So if you have a wide bridge nose and you need an extra wide glasses bridge, for example, that's contributed to heavily by the shape of your maxillae. The inferior half has the lateral and alar cartilages, and these are the cartilages that give the bottom part of your nose its shape. And the ala nasi is the flared portion that forms the nostril. So the little bowl-shaped flared area, and it's shaped by dense connective tissue primarily, which forms the lateral wall of each nostril. So here are all of these things respectively. So we've got the nasal bones. So these give your nose the profile it does, and then here are the portions of the maxilla that contribute to overall nose shape. You have a septal cartilage, and this is part of the cartilage that divides your nose in half. And then you have lateral cartilages, which help support and hold your nasal passage open. The greater alar cartilages, there are two, one on each side, and those are what give your nose tip its shape. And you have lesser ones as well in the corners here. And then there's dense connective tissue, primarily irregular, that forms the nostril shape. So if you're going to get your nostril pierced, your piercer is going to shoot for in here. And if you're going to get your septum pierced, like mine is, the goal, because the nasal septum does descend down here, is to pierce the nasal septum through the non-cartilaginous part, which is just under here. So the nasal cavity, which is the total space inside of your face, extends from your external nares, or nostrils, to the choanae, which are the posterior nares. So because your nasal septum divides your nasal cavity in half, you not only have two anterior nares, but you also have two posterior ones as well. So the ethmoid and sphenoid bones compose the roof, and the palate forms the floor. And of course, one of the two parts of your bony nasal septum is the vomer, and the other is the perpendicular plate of the ethmoid bone. So the vestibule is the dilated chamber inside the ala nasi, which is the area surrounded by your nostril. And this contains stratified squamous epithelium, not keratinized, and guard hairs, which prevent particulate matter from making their way into your nasal cavity. The septum divides the cavity into right and left, and those chambers are called nasal fossae. The anterior part is formed by the septal cartilage. The inferior part is formed by the vomer, and the superior part is formed by the perpendicular plate of the ethmoid bone. So if somebody has a deviated septum, that means that one or all of these elements have been compromised either by being perforated or being crushed in one direction or the other. So this is the head in sagittal section, and you can see some other elements of the nasal cavity here as well. So you have the superior and middle nasal conchae, that means shell. So these are bony protuberances covered with flesh, and this is designed to make airflow through your nasal cavity not laminar but turbulent. Here's the vestibule. Here's the inferior nasal concha. Now notice you're not seeing the perpendicular plate of the ethmoid bone or the vomer because this is a little bit left or right of sagittal so that you can see the cavity as a whole, as opposed to just seeing the septum. So what do I mean by turbulent? Well, if air zipped back through here this way and went more or less straight, it would not have time to be cleaned or to be moistened or warmed up by the nasal cavity. So you would get dry, dirty, and possibly cold air entering your pharynx. When you force the air to swirl, which is what the conchae are for, the air has time to come into contact with the wet walls of your nasal cavity before it makes its way down your pharynx, and that gives it a chance to get moistened and get warm and have particulate matter cleaned out of it. So you're actually designed to breathe through your nose, not through your mouth. Humans resort to mouth breathing when we're trying to really gulp air out of the environment. It's not really for quiet, passive breathing. Here's another view of the same thing, but this isn't a real human cadaver. So this is a dissection preparation that is sometimes done, not always. Not always because it compromises the ability to view other structures, but if you have enough cadavers, you can kind of do this. So here you can see the superior and middle nasal conchae as well as the inferior one. Here you can see the cribriform plate of the ethmoid bone, so you know that the nerves of the olfactory bulb are going to stick through here. You can see the posterior nares or choana. And you can see here's the nasopharynx right here. There's the uvula, that's the little dangly bit in the back of your mouth, and so on down. All right, so the choanae and the meatuses. The superior, middle, and inferior nasal conchae are folds of tissue overlaying some bone, and those mucous membranes are, of course, supported by those bones. We call these turbinate bones because they look like a turbine, which is a spirally structure that is designed to move air, or in some cases, of course, water. And the meatus is the narrow air passage beneath each concha. This should say concha. That's plural. This is singular. And the narrowness and turbulence, as I mentioned, ensures air contacts mucous membranes instead of just cruising right on through. The mucosa of the nasal cavity is also special. So the respiratory mucosa lines the nasal cavity with ciliated pseudostratified epithelium primarily. So this is a very common epithelium. You'll also find it in the trachea and bronchi. We're going to keep seeing this kind because it's good at moving mucus around. But the olfactory mucosa, as you saw last term in the microscope in the lab, lines the roof, and this includes things like Bowman's glands and other stuff that is there specifically to facilitate smelling rather than just breathing stuff. The mucosa also has a defensive role. So mucus helps trap inhaled particles because bacteria and other particles, even if they're not alive, have a hard time moving through a thick gelatinous goop. They can't really swim or diffuse through it very easily, so that kind of stops it in its tracks. It's like a glue trap for bacteria. And then if there are bacteria, as opposed to just dust or what have you, in the mucosa, lysozyme is secreted by nearby cells, and lysozyme causes cells to lyse, and it is an enzyme, so that's why it's named that. All right, the cilia and erectile tissue. So one function of the nasal cavity is to move mucus around, and that is, of course, achieved by the presence of cilia on the respiratory epithelium. So these take that debris-laden mucus into the pharynx to be swallowed. So above the pharynx, your cilia beat down and back to move that stuff down. And below the pharynx, the cilia beat up and forward to move it up from the trachea into the pharynx so it can be swallowed and enter the digestive system, which has special acids and enzymes to deal with all that stuff. You also have erectile tissues of the inferior nasal concha, particularly, so here we see the singular form is preserved and correct. Well done, Ed. So these are venous plexuses. So remember, a plexus is a braid-like network, and these happen to be venous rather than arterial, which is interesting because we only really covered alterations of the arterial system last term. Venous ones are fairly rare, but they do exist. So these ones rhythmically engorge with blood, and it shifts the airflow from one side of the fossa to the other about once or twice an hour to prevent drying. So you may have noticed this if you've ever been really sick and trying to sleep and one side of your nose closes. That's because your nasal mucosa is already inflamed, and the venous plexuses are just sitting there doing their jobs, but because your interior nasal tissue is already puffy, that rhythmic engorgement ends up closing off one half of your fossa where it wouldn't otherwise do that. So if you've ever been plagued with a cold and laying in bed trying to sleep and wondering, why won't my nose behave itself? That's why. Spontaneous epistaxis, which is called a nosebleed, is usually present in the inferior concha, and this is because the epithelial tissue inside the nasal cavity has developed a thin spot, either from you picking your nose or booger flakes off and thins it out or something of that nature. And then the venous plexus is exposed to the air and bleeds a little bit, and they usually resolve themselves pretty quickly. I'm not sure if you can hear my cat screaming at me in the background, but he is. So if you can, I'm sorry about that. He's desperately soliciting my attention. All right, so we need to get to the regions of the pharynx. And fortunately, they're named for their location. So they're all part of the pharynx, which is a muscular funnel that's designed to transport liquids like beverages, food, and air. And the regions are named for where they are. So the nasopharynx is at the back of the nasal cavity. The oropharynx, as you can see, corresponds to the posterior of the oral cavity, or your mouth. And the laryngopharynx, laryngo might be a less familiar prefix, corresponds to your larynx, which is a cartilage box that is the entrance to your trachea. So, note, we've got some differences in epithelial tissue between various areas of the pharynx. No, Orange, you cannot have my stylus. Go away. So the nasopharynx is filled with and lined with pseudostratified epithelium. And again, that's so that you can move that mucus around and get stuff out of there. So posterior to the choanae and dorsal to the soft palate. So if we go back here, we can look at that. So here's the choana, and here's the end of the soft palate. So this is where that is occupying. It also receives the auditory tubes, which is why you can change the pressure in your nasal cavity while blocking your pharynx to change your ear pressure. Think about the last time you maybe tried to equalize your ears on a plane or when you swam really deep in a swimming pool. It also contains the pharyngeal tonsil. And the air turns 90 degrees downwards, which helps to trap large particles. And large here, we're using that term relativistically. That means about greater than 10 microns or so. The oropharynx is like the majority of your oral cavity, stratified squamous epithelium. And this is the space between the soft palate and the root of the tongue. It goes inferiorly as far as the hyoid bone, which remember is that weird little floating bone in your throat. And this contains your palatine and lingual tonsils. So your palatine tonsils are easily viewed on either side of your oropharynx laterally, and the lingual one sits on the very back of the root of your tongue. The laryngopharynx is also stratified squamous epithelium, and this is from your hyoid bone down to your cricoid cartilage, which is a cartilage you'll learn a lot more about in lab, excuse me. And this is the bottom of your larynx, right before the trachea. Okay, so speaking of the larynx, you may have heard of the glottis, and the glottis is the name for the superior opening of the larynx. And I want to stress this because it's something that's students struggle with it in lab because it's sort of like a foramen in bones where it's not a structure but rather an absence of tissue, so it's an opening or a passage. So this is the opening or passage that admits entrance into the pharynx. I hope that makes sense. It's hard to describe without something to point at. Now, sometimes you may want to swallow something and direct food and drink to the esophagus. That's the tube that's for eating and drinking, not your trachea. That's the air tube, not the food tube. This one is the food tube. So you have an epiglottis, which is a spoon-shaped flap of tissue. It has elastic cartilage inside of it, and it's covered over with epithelium, and this thing tips down and covers the glottis while you're swallowing so that food and drink go down the right tube. Now, if you try to achieve the impressive feat of talking while swallowing, you may disrupt the epiglottis in doing its job and begin to choke. Interestingly, in infants who have different feeding and laryngeal needs than we do, the larynx is actually higher up in the throat, and that's related to tongue and pharyngeal musculature. And what this allows the infant to do is form a continuous airway from the nasal cavity that allows them to breathe while swallowing. And this is really designed to facilitate breastfeeding. Oops, whoa, what happened there? So if you are in this class because you want to become a lactation consultant, for example, oh, I figured it out. If I push on this, it advances the slide. Got it. Yeah, so if you're here to be a lactation consultant or some such, this is a piece of information that you might find really useful. Or if you're just a mom or aspiring mom and you're wondering, why is my baby having a hard time breastfeeding? This might be why. There might be something going on with this. There could be a lot of reasons. So by age 2, the infant develops a more muscular tongue because presumably they've been learning to talk, which tones up the tongue as well as manipulate solid foods, and that ends up forcing the larynx down just by virtue of an increase in muscular tone. All right, so here are some views of the larynx. And I want to stress that these two views right here have had all of the flesh stripped away, so you're really only seeing the ligaments, bones, and cartilage. So here is the epiglottis. As I promised, it is spoon-shaped. So this is the side view of it, and this lid here tips down when you're swallowing so that food goes down the esophagus, not down the trachea. Here's the hyoid bone. It's connected to the thyroid cartilage by the thyrohyoid ligament. This is the thyroid cartilage, so it forms the bulk of the cartilaginous box of your voice box. And the laryngeal prominence is one of the sexually dimorphic traits of humans. So XY individuals have testosterone receptors right there, which develop into the quote-unquote Adam's apple, and XX individuals tend not to, so they don't tend to develop that laryngeal prominence as much. Here's the cricoid cartilage. It's a ring-shaped piece of cartilage right before the trachea, which is everything down here. And here's the back of the whole shebang. So you can see the cricoid cartilage, and it connects to two pointy pieces called the arytenoid cartilages, and those are tipped with the corniculate cartilages. So you'll learn those in lab. All right, so the walls of the larynx. The interior wall has two folds on each side, from the thyroid to the arytenoid cartilages, and these are called the vestibular folds and vocal cords, respectively. So the vestibular folds create sort of a web-like fleshy closure during swallowing, and I'll show you a picture of this. And the vocal cords are medial to the vestibular folds, And these are the ones that you use to produce sound. So I'm doing that right now. You do this by passing air past them and then using your muscles and your arytenoid cartilages to change the length and tension on your vocal cords so that you can make very high-pitched sounds or very low-pitched sounds, depending on what you want to do. So the intrinsic muscles are what they sound like. They're inside. And these rotate the corniculate and arytenoid cartilages, and that adducts or abducts the vocal cords. So adducts means tightens. So it turns out when you rotate them inward and adduct them back towards the medial line, that tends to make them tighter and thinner and produce high-pitched sounds. When you relax those muscles and abduct them, that allows you to produce low-pitched sounds. And that is all achieved by acting on the vocal cords. Extrinsic muscles connect the larynx to the hyoid bone, and these are more for swallowing. So if you put your hand on your throat while you're swallowing and then you do a big gulp, you can feel your hyoid bone tip upward and then back down, so you can feel those extrinsic muscles working. So I mentioned I would show you a picture of it. So this is a relaxed glottis, which, considering this person literally has a camera shoved down their throat, is pretty impressive that they're able to do this. We'll show you a video in class of somebody singing while having a camera in their throat, so you can actually see that the way that their vocal cords behave. It's pretty cool. So here's the epiglottis. I'm going to outline the glottis itself with my pen. So remember I said it was an opening rather than the presence of something. So this space that I'm outlining with a dotted line is the glottis, which is the opening onto the trachea. Here, you can see the two corniculate cartilages, and then down here are the arytenoids. So these rotate inward and outward, so this way and this way, to place different elements of tension on the vocal cords. So here is one vocal cord, and here is another. And then this web of tissue that covers the rest of the opening, that's the vestibular fold. So you'll sometimes see these called false vocal cords because they look like they're for sound production, but really it's these guys that do the sound production. Okay, so let's look at some adducting and abducting. If you adduct, that means you're going to swivel those arytenoid cartilages in closer to the midline, and that's going to bring your vocal cords together and also make them thinner or narrower. If you abduct or let them relax outward, you're going to have two thicker and further apart vocal cords, which allows you to produce lower sounds. So those are the basic mechanics of how you can change the pitch of your voice. And of course, the speech component involves making your mouth into various shapes and then passing air through the shapes. Speaking of speech and whispering, so speech is modified sound made by the larynx. So you send sounds through your larynx and make different tones of things. That's why I am speaking non-monotonously. I can change the tone of my voice to perform different inflections and sound like a human instead of sounding like a robot. So speech requires the pharynx, the mouth, the nasal cavity, as we discussed, because remember, if you pinch your nasal cavity shut, here, I'll do it for you. You sound like this, which sounds really, really different from my normal speaking voice and probably really annoying, so I'll stop now. Your sinuses also contribute to resonance of your voice, and this gives us a wonderful diversity in voice types. So the tongue and the lips actually form the words, and depending on, where you grew up and how finely you're able to control your tongue and lips, you're going to have different accents and place emphasis on different phonemes and parts of words and have different styles of attack, which is the start of a sound you make. And that gives us the wonderful diversity of accents and speaking styles and language that we've got, which is really delightful. I'm super glad that not everybody speaks the same. So pitch is controlled, as I mentioned, by tension on the vocal folds, and when pulled tight, those produce a higher pitch. Male vocal folds are thicker and longer, so they vibrate more slowly, producing a lower pitch. And I'm going to cross this out because this is not quite correct by modern standards. What we mean here is XY folks that have chosen not to undergo any kind of hormonal treatment. So if you want to whisper, if you want to do this, you're forcing air through the almost closed rima glottidis, which is the small opening between the arytenoid cartilages when your glottis is closed. And your oral cavity alone is forming the speech, which is why it sounds kind of like wind talking. Okay, so we're back, and sorry, I had to take a break and take a drink because me forming speech was challenging without water. Alrighty, trachea. So this is your breathing tube. It's a rigid tube. It's only about 4.5 inches long, so people think it's a lot longer than it actually is. It's pretty short. And it's about 2.5 inches in diameter or so. It's in front of the esophagus, and this is significant because this is the reason for the C shape of the cartilage rings. So we call them rings, but that really should be in quotation marks because they're not full rings. They don't go all the way around. They are shaped like a C. And the opening in the rings faces posteriorly towards the esophagus so that when you swallow a bolus of food, it's able to pass by your trachea without pain. So you have a trachealis muscle in the back that allows your trachea to flex when food goes by and allows airflow adjustment by expanding or contracting. So the larynx and trachea are lined with ciliated pseudostratified epithelium, which functions as a mucociliary escalator, which again is one of my favorite anatomy terms. So mucociliary means mucus plus cilia, and escalator, of course, means to move up. So that's how that works. So contributing heavily to those two things are the cilia. So here they are in yellow. Now, again, not really yellow. This is a scanning electron micrograph. And the mucus is produced by goblet cells. So this is a bird's eye view. So you're looking down on the epithelium, so you can see the top of the goblet cells here, as well as their cell junctions. So see these little red areas? These are the tight junctions and other cell junctions that allow the cells to connect to each other and prevent bad guys from sinking between your cells and going attacking deeper tissues. So we can directly see the consequence of cell junctions just in this picture, which is pretty nice. Okay, so now we've cleared the upper respiratory tract entirely, and we're going to descend to the lower respiratory tract. So the lower respiratory tract has the trachea and bronchi, and the bronchi and bronchioles are an increasingly smaller series of tubes, and the histology of this region changes the further down you go. But the bronchi and the trachea have the following in common. There is a ring of hyaline cartilage. There is pseudostratified ciliated epithelium lining the lumen. In the trachea, there are numerous glands, which produce lubricating fluid. And you're also going to see a lot of goblet cells in the epithelium to contribute to mucus production. So the primary bronchi have C-shaped rings, just like the trachea, and these arise from the trachea after 2 to 3 centimeters. They enter the hilum of the lung. So if you look at this, trachea goes down, splits into the left and right primary bronchi, and then there's actually a little bit of bare bronchus before the lung starts. I'm going to draw an imaginary lung outline here. There we go. The right bronchus is ever so slightly wider and a little bit more vertical. And so it tends to be the case that if something is aspirated, like a bit of food or some such, it usually ends up in the right bronchus rather than the left. The secondary or lobar bronchi have not C-shaped rings, but rather overlapping plates. So these kind of look like this. So a plate of cartilage, and maybe there's another one over here. So it's not a C shape, but rather just some plates. So these branch into one secondary bronchus for each lobe of the lung. So a lobe is a histologically and anatomically discrete area of the lung. So if you, let's say you need to get an area of your lung removed, they can't just lop off a piece of it because that would leave a bunch of alveoli exposed to the air. So instead they have to go in and do a lobectomy where they take out an entire lobe of the lung. A tertiary or segmental bronchus, so bronchi is plural, bronchus is singular, and I'll write that up here because like with vertebrae versus vertebra, pluralization and singularization of these words have historically been a problem, specifically on practicals in lab where spelling extra super counts, so I want you to be mindful of your plurals. So you've got 10 right and 8 left segmental bronchi. And when we get to lung anatomy, it'll become a little clearer why those two things are. And then a bronchopulmonary segment is the portion of lung supplied by each tertiary bronchus. So this is a picture where each bronchopulmonary segment has been colored a different color. So let's say I'm a surgeon and somebody has some cancer down here. It would be real easy for me to just cut this way, right? But then all of the alveoli along this cut line would be exposed to the air. So instead, what I have to do is go in and cut this one, and I have to seal it up, and then take out all the tissue that is associated with that one. So that is how that is generally done, instead of just cutting straight across. But you can see this is a primary bronchus. Here is the corresponding secondary bronchus. And then off of that, there's a third. And here's a third, etc. So bronchioles are different from bronchi. So remember, eole means the tiny itty bitty version of something. These lack cartilage and they have instead smooth muscle. All right, so bronchioles have a layer of smooth muscle. Oopsie. Wow, what have I done? I put my thumb down on the wrong spot. Sorry guys, still getting the hang of this Microsoft Surface thing. Well, I will. All right, sorry about that. I got a Surface so as to better screencast for you, but I am still figuring it out. My bad. So we have a layer of smooth muscle, and that helps them to contract and dilate according to need. So if you have asthma, that sometimes means that in response either to allergens or to exercise activity or a variety of other things, your bronchiolar smooth muscle just spontaneously constricts super, super tiny, which makes you feel like you're breathing through a straw. And that's very uncomfortable and in some cases extremely dangerous. So that's one of the reasons for that pathology. A pulmonary lobule is the portion of the lung ventilated by one bronchiole. And each of those divides into between 50 and 80 terminal bronchioles. And the word terminal here means end. So this is the end of the conducting division. So the terminal bronchioles have cilia, but they give off 2 or more respiratory bronchioles. So how can something be a respiratory bronchiole? How can it be both a bronchiole and contribute to gas exchange? Well, these little guys have alveoli hanging off of them. So they divide into alveolar ducts. Alveolar ducts end in alveolar sacs, and alveoli bud from the respiratory bronchioles, alveolar ducts, and alveolar sacs. So let's look at a picture of this before we go back to the previous slide. So each of these little air spaces here is an alveolus. An alveolar duct, you can see bracketed right here, is basically a hallway made of alveoli, so I'm drawing lines inside of a bunch of alveolar ducts. And here you can see bronchioles, and this one happens to open into this alveolar duct, making this a terminal bronchiole. And you can see the same thing in a scanning electron micrograph down here. So the histology, as I mentioned, changes from pseudostratified ciliated columnar to non-ciliated simple cuboidal as you pass deeper and deeper into the lungs. And at the end, the very end, you have alveoli, which are simple squamous. And of course, they can't really be anything else because how else would you accomplish gas exchange? You need the membranes there to be very thin. So Incomplete rings of cartilage are gradually replaced by rings of smooth muscle and then simply by connective tissue, which forms the underlying floor to which the epithelium sticks. So, sympathetic nervous system and adrenal gland releases, including epinephrine and other catecholamines, that causes relaxation of the smooth muscle and dilates the airway, which is a little bit counterintuitive because we tend to think of epinephrine as being a tension generating or tightening neurotransmitter. But in this case, if you're preparing for fight or for flight, you want to increase oxygen availability, so you're going to want to blow open those airways. An asthma attack or an allergic reaction, such as anaphylaxis, constricts the distal bronchial smooth muscle, causing you to feel like, as I mentioned, you're breathing through a straw. So nebulization therapy means that you inhale mist. So nebulization is turning something into mist, typically drugs. So albuterol, for example. Let's write that in there. I-E. And albuterol is a steroid that helps to relax that smooth muscle, and also other drugs can reduce the thickness of bronchial mucus as well, and that facilitates breathing. So here's the tissue of the lung once again. We've got an alveolar duct, which is just a little hallway made of alveoli, which I'm outlining here. So you can see it's a tube made of alveoli. And then down here, this is probably the alveolar sac that corresponds to it, which is basically a little cul-de-sac of alveoli. So there's a blind end. And then here we can see the terminal bronchiole. The respiratory bronchiole is about here. And here's another view of the same thing. And here we have included the capillaries. So note that each alveolus has its own little capillary bed, and you can actually see gas exchange happening. So this is going to be a branch of the pulmonary artery or vein. In class, I will ask you to tell me. So if you know anything about the pulmonary circuit, you'll know that this is a branch of the pulmonary artery because arteries carry blood away from the heart and towards the lungs, in this case. And the blood is going to return to the heart via the pulmonary veins. So we call this one a venule. Now, if we zoom in on the alveolus themselves, or the alveoli themselves, excuse me, at the capillary interface, you can see we have a squamous alveolar cell. So here is the nucleus, and then this is the rest of the cell. So this is one of the cells making up the wall. Here's another one. So these are really thin, so that O2 can easily get in here, and CO2 can easily go into here. Now you have two other kinds of cells that make up the internal structure of the lung tissue. You have an alveolar macrophage. So these are going to patrol your alveoli and eat dust, which does get into your lungs, and particulates, which is not dust but other small particles. And bad guys if they get in there, so bacteria, for example. Great alveolar cells, which are also sometimes called septal cells, these produce a fluid called surfactant, and surfactant is this stuff. Basically, surfactant is a mild detergent. It disrupts the surface tension of water. And that's because you don't want the walls of your alveoli sticking to each other. That would make it really hard for you to ventilate your lungs. So cell types of the alveoli. Type 1 alveolar cells are the ones that make up the walls. So these are simple squamous by definition because they have to be nice and thin in order for the gas exchange to occur. Type 2 alveolar cells are also called septal cells, and these ones are covered on the free surface by microvilli, and they secrete that surfactant that helps you to inflate your lungs and resist the surface tension of water. And the wandering macrophages are also called alveolar dust cells because dealing with dust is kind of their primary job. So, speaking of the alveolar-capillary membrane, the respiratory membrane is about 1/2 of a micron thick, which is very, very small. So to put that in perspective, a red blood cell is about 6.5 microns in diameter. This is 1/2 of 1 micron. These facilitate the exchange of gas from the alveoli to the blood. And there are 4 layers of membrane to cross, which include epithelial wall of type 1 cells, the basement membrane of the alveolus, the basement membrane of the capillary, and the endothelial cell of the capillary. So all of these make a big sandwich, which is about 0.5 microns thick. And together, if you were to flatten out all of your alveoli, so all of your respiratory membranes, the surface area would be incredibly vast. So it would take up about the size of a handball court, which is really, really, really big. So that is how you get maximum gas exchange capability while not having a super big thorax or some sort of weird sail-like apparatus where you wave your membrane in the wind and collect gas that way. So it's a way to make gas exchange efficient while also carrying it in a relatively small package, aka your thorax. So that's where we're going to stop for now, because it gets hard to upload these things to YouTube if they get any longer than that. And hopefully the next one I will have all the kinks worked out of how to use Snagit, the software I use, with the tablet mode, and so we won't get that slide flipping thing that we had this time. I apologize for that. All right, see you next time.